All right, so what is LabKey's Biologics LIMS? It is an integrated suite of tools that centralizes and aligns sample, molecular entity, experiment data, and notebooks. Really, it's a combination of many powerful components of LabKey's existing product suite, including everything you learned about Sample Manager yesterday, plus unique components specifically designed for biologics research and development. It is built on LabKey Server, making it a highly adaptable system suited for wide-ranging use cases. I'll have the chance a little bit later to dive into more of those specifics during my demo. It was designed with ease of use in mind, ease of use and adoption in mind. Those are some of the main themes that you heard our CEO talk about yesterday as being major product themes at LabKey. We know that organizations need solutions that can be deployed rapidly without undue barriers to usage. That is why having a cloud-based product is so important. You do not need to make a large technology investment to begin getting your data organized. You also do not need a deep IT or informatics team. We have a very experienced support team prepared to train up your scientists and your administrators and guide you through implementation. Our Biologics LIMS is, as the name implies, oriented around biologics development. It starts with recognizable data structures and entity relationships common to the industry, such as nucleotide sequences, vectors, constructs, molecules, etc., but leaves you the ability to both update those data structures and add your own. Each of these registry structures relates to the samples that flow through your teams, enabling propagation of critical information as those materials progress through your pipeline. All right, LabKey's Biologics LIMS comes in two product tiers. Our starter offering is really oriented towards early stage biotechs, academic labs, not-for-profit groups, and other small organizations looking to invest in tools to help them get organized. It's a cloud-based offering and offers rather significant value, even for just a few users. Comparatively, our enterprise offering is aimed at organizations with a more mature informatics architecture and can be integrated programmatically into that broader landscape. It's for those organizations really looking for a central source of truth for their data that still supports system extensibility. We are fortunate enough in a few minutes to be able to hear from our very own Adam Rauch, who will take a bit of a deeper dive into what that means. I'm going to briefly show you this image again, which many of you saw yesterday from our LabKey presenters. The concept I want to emphasize here, which was also emphasized by our CEO yesterday, is that you can grow with LabKey. I just finished outlining those Biologics LIMS tiers, and they provide a seamless path up through that powerful tooling available in Sample Manager that Hannah walked through yesterday. If you liked what you saw, but wondered how to tie in your molecule, cell line, protein sequence data, our Biologics LIMS is that next logical step. If you're unsure, you can start with the basic functions you need now and upgrade seamlessly as you scale. Okay, so as with all of our products, Biologics LIMS was developed in partnership with our customers and our friends. We get to spend lots of time talking to them and learning from them, and that's really valuable and and satisfying. We've learned the myriad ways in which efficiency is so highly prized, and that when there are too many hurdles in the way of speed, organizational adoption and even employee satisfaction can be affected. So to minimize these hurdles, we target a number of organizational problems. First and foremost is going to be that scientist buy-in. Now, we're all forced to learn spreadsheets at some point, which are, of course, very powerful. And picking up alternative tools must be at least as easy to use, which is why we at LabKey, of course, have an entire team of user experience designers and ex-scientists to help guide our products. Those new tools also need to serve a clear purpose. They need to ameliorate common pain points in the industry. Spreadsheets can solve lots of problems, but they are just about the least built-to-purpose tool out there. They also have to scale. Something that serves a scientist a few at a time, but falls down at a larger scale, is simply going to become a burden. Similarly, something that serves those individuals reasonably well, but doesn't serve other members or teams at the institution simultaneously, is also going to present some downstream hurdles. Then there's data standardization, and I'm going to add conventions to that. If the way I capture data and metadata about a vector or cell line suits me nicely, but it's missing critical information or is in a difficult format for the team I'm passing data to, it's going to slow down our collaboration and maybe the whole process entirely. It's going to really also annoy your data scientists later. Driving agreement across your organization for data structural expectations and then enforcing those expectations really levels those hurdles to collaboration and progress. As an added advantage, it will help deepen your systematic thinking about data and collaborations, as well as one scientist or one team of scientists understanding of how other teams work, what they expect, what they need from you, or what you need from them. Then there's governance and access controls. This is really two main components. So at some point, right now you may just be 5 people working in a lab, but at some point you're not actually going to want everyone to see everything. With limited exceptions, you will need a way to organize your data and impose a permissions model based on your business needs or other incentives. That's security. Improved focus is really the other benefit. That organizational structure is going to make it so that when you go looking for information, you don't have to sift through all of it, unless you want to, of course, to find what you're looking for. You can filter out work that's part of projects that you don't particularly care about right now. Then there is collaboration tools. There are a variety of other forms of communication besides the data itself that are critical. Scientists need to convey previous work, results, critical conclusions, notebooks, methodological guidance, and things like requests of each other. And having all of those in one system, in the same place as your data, is highly enabling. And then lastly on this slide is data centralization. And while data centralization, while centralization of data from disparate systems is in and of itself one of the most potent improvements to collaboration, there is quite a bit more value here. When the expectation is set that your clean data will reside in one system, trust in that data is going to improve. Tracking or auditing of that data becomes realistic, and the ability to troubleshoot and discuss problems with those involved becomes simpler. This also helps prevent many of the painful effects of turnover, like you heard from Dylan yesterday at OSU. As people cycle through your organization, their knowledge and accomplishments can be maintained. You won't find some huge mass of files in one particular place that may or may not be full of critical information that you can no longer process. It's all in one place in an expected form, and it helps maintain that that traceability and availability. All right. So when you do get our Biologics LIMS implemented, there are many significant benefits, some of which are clear at the surface, and as I mentioned on the previous slide, others are deeper and more systemic. Biologics LIMS really helps organizations ensure data uniqueness, traceability, and availability. Why is that important? How much extra work might you avoid if you are told a cell line you're experimenting on already exists with lots of data when you try to register it, or that some molecule already has an established purification method when you get samples of it, instead of going and redoing all of that work? How much time and brain power do you have to put forth capturing the vector that was used to produce every single sample that you have in your spreadsheet, or whatever system that you happen to be using? Making sure that registered entities are unique, that their relationships with everything else in your system are traceable, and that data is presented to you, that data is available, is what saves all of that effort. And as I said on the last slide, a prerequisite for this advance is that standardization and shared expectations. Let's see. Then there's integrating samples, entities, and related data. There are some very practical, everyday benefits of having your data centralized, but also integrated. So managing samples at one or multiple sites is something that can become streamlined, which you saw in Hannah's sample manager presentation yesterday. Your regular data stand-ups or compiled PowerPoint presentations for the purpose of handing data off between teams, between scientists, between sites, require no or much less preparation, because you can simply point to it in the system. And lastly, general exploration of existing data for project management or project planning is no longer so labor and time intensive. You don't have to check 5 different systems and dozens of files, perhaps in many locations, in order to accomplish those things. Then there is connecting data for querying and compilation across teams. I guess I'm going slightly out of order, but that's all right. Another effect of the previous point is that having all of that data connected means that you can query across it. You can ask questions of that data that is connected, including data produced across multiple teams. It just becomes so much simpler. Before, you might have had to hire Sherlock Holmes at some kind of obscene hourly rate to help you produce a client report or a regulatory submission. But with our Biologics LIMS, you can let that detective atrophy in his parlor, which is good. Then there's authoring notebooks connected to structured data. You may have heard at a previous webinar or in Hannah's presentation yesterday, the phrase data connected ELN. Notebooking I would categorize with death and taxes. This is totally inevitable. You have to document your work. It's a fundamental component of the scientific operation. And if you have to do that every day, I would strongly recommend that you not lock critical information into unstructured notebooks. It makes it very difficult to solve some of the previously mentioned problems, and many other forward-looking issues that you're likely to run into. If you make use of LabQ's Biologics LIMS, make use of our data connected ELN, all of that compiled and connected data that I have been talking about can be referenced in your notebooks with just a couple of keystrokes. And I promise that having your data structured will also prevent myriad problems for your data scientists, among others. as you grow. So we know that your organization is constituted of many scientific specializations. Whether you're 5 or 50 or 500 people, we all specialize for that comparative advantage. But that specialization does not need to lead to the crippling siloing of data. So take those gains from the division of labor, but don't lose the value of having your data integrated. Looks like I have a couple of questions that come in here. Let me actually hit my next slide before answering a couple of these, because it seems like some of the questions are about implementation. I think it'll be helpful to better understand some of the feature areas before diving into that. So you might have heard enough from me to predict the composition of our biologics LIMS, but allow me to briefly outline it for you. These, as you can see on the screen, are a couple of the key feature areas. They include sample management, and this is what you saw in the demo yesterday of our sample manager product. All of those functions are available at all of the tiers of our biologics LIMS, and those sample manager functions allow you to track lineage and connect samples to related samples, experiment data, and entities in the system. You get to define all of those different sample types. As you heard from Sydney at OHSU yesterday, they had a variety of sample types, and I heard somebody ask about why they chose the ones that they did. You get to define all of those types. You get to define their metadata. You can capture the lineage within those samples, examine the chain of custody and history of those samples and their locations, and manage your samples in freezers, your sample inventory. This, of course, closely relates to the next feature up there, which is our bioregistry. Here, you can define and track the various entities that are important to you. This includes registration of molecular entities, such as molecules, several sequence types, vectors, constructs, compounds, plus any others that are relevant to your research. Many of those come pre-built in the system, which we allow you to make some modifications to. But of course, you can add your own and build lineage relationships between these entities and your samples, enabling activities like the project planning based on existing related experimental data. Then there is our workflow manager, which helps you create and manage tasks and make requests of each other. Those processes are standardizable and can be built with all kinds of tasks that have comments and files, assay results, sample information, and other metadata. I would say the most common way this is used is for making and managing requests for assays and analyses, but it's used in a variety of other ways as well. Similar to our bioregistry, our media registration area enables you to register and track the raw materials, ingredients, mixture recipes that you are making regular use of, and produce batches, which are trackable like the other samples in the system, to help make your media preparation consistent. Then there's our electronic laboratory notebook, which I mentioned a little bit previously, that speeds up your documentation of experiments through an intuitive and data-connected interface. As I mentioned before, use of all of our other data capture and data relationship mechanisms allows you to quickly enrich those notebooks through direct data referencing without, this is the important part, having to compile that information again. You can really cut down on time spent authoring by making use of full fidelity templates and further simplify your lives with collaborative authoring. My favorite example of collaborative authoring is what I see often done by teams of cell line development scientists, who is part of, say, a single epoch of work operating in shifts to care for their cells, keep them alive, passage them, take measurements of their viability and density, things like that. And in our system, you can compose your portion of that work stamped with the different days you were involved, and have it all in one place, request only a single review of the whole document, and really streamline the process. And last but not least is our assay data framework, which allows you to capture experiment data, instrument data, and automatically connect it to the samples in the system. And with all the data in one system, looking across experiments for insights or to make decisions about your materials becomes a lot easier. Okay, with that, I do see a number of questions. Let me give them a quick read here before I move on and see if now is a good time to answer some of them. Okay, so there's some interest in how, in what an implementation looks like. So the way implementation works is that when you work with us, you receive a primary representative from our client services team. That's some kind of technical account manager, as well as all of the other members of the team that support them. This account manager will work with you to understand the way that you currently store, manage your data, and will help you model it in useful ways within our system. So there's an exchange of information, there's some experimentation and show and tell. We work up a plan for getting all of your data structures designed, start getting data into them. We also offer comprehensive training on the way that all of our products work. So we can define how each of your different types of scientists or administrators are going to make use of the system, prepare each of them, and implement each of those as we go along. Let's see. I see. Can the bioregistry handle constructs containing multi-enzyme pathways and complex strain genotypes with multiple SNPs and indels? I think it would be helpful to tear that apart a little bit individually. When I show you more about the bioregistry and the way that we handle sequences, some of this will become a little bit more clear. But we allow you to capture both nucleotide and protein sequences in our bioregistry. Each of those, of course, are kept unique, and we allow you to annotate those with different sequence parts, which we maintain as those sequences are used to produce proteins of various types. Let's see, we track their insertion into the constructs used. Those constructs are related to the cell lines that they're transfected into, and it's all well connected. I see another question here. For your more production-minded customers, does the Biologics LIMS interface with liquid handlers and automated systems? Can it create work lists and import-export data from the liquid handler based on samples and workflows? So we have a variety of faculties for receiving and producing sample manifests, things like work lists. We have people that use our system in communication with systems like that, but we are always eager to make those more efficient, easier to set up, things like that. So maybe the asker of this question, we could spend a little time digging into exactly what your particular work lists look like and how it would be useful for that to flow in or out of the system. Okay. I see another question here. I guess I should talk faster, about a small group, maybe not quite ready for everything that I've described. So what I would recommend, and I could jump all the way back to that growth slide that I talked about, is we will help scale with you in terms of number of users, in terms of functionality. One of the things that I think is most important about our product suite is the way that they grow into each other. So if there are certain facets of our products that are of value to you, and others that are not, it's an almost sure thing that one of our products contains what you want and excludes the things you don't currently need. And as your science develops, as your number of people and the complexity of your operations grow and you need those other things, we allow you to seamlessly upgrade through those tiers, making it really easy to scale. And then I see an inquiry about some of the ways in which our Biologics LIMS is extensible. I think that's a perfect question for our next speaker. So let me jump forward a tiny bit and invite you to connect with us. So I think my favorite part of this job by far is having conversations with researchers and organizational leaders trying to improve how they and their people do their work. I really love to hear more from you about your needs, industry trends that you observe, things that affect your decision making. And I'd be glad to dive deeper into these features for you, with you, even if it's currently a hypothetical thing. Like in the future, this is something we want to do. What will that look like in your system? We would love to field those questions.